And all right, so yeah, I'll be talking to you all today about the acute and enduring effects of hallucinogens on brain function and cognition. So first of all, what's a hallucinogen? So if this talk were done in person, I'd be surveying the audience, and chances are nobody would give me a really satisfactory answer. And that's because there are several different types of hallucinogens that we kind of just group together. And the reason I think we group them together is because these are all drugs that don't neatly fall into uppers and downers. So you have your classic psychedelics. These are the serotonin 2A agonists. This is what I refer to as a psychedelic. I save this for serotonin 2A agonists. I realize that other people don't do that, but this is what I do for this particular purpose for this talk. And so psilocybin, LSD, 2CB, etc. They're found in nature, such as in psilocybe mushrooms, as well as I just found out that pretty pure-ish quantities of bufotenine and not other psychedelics are found in the seeds of the Sibyl tree. Then you have your NMDA antagonists. These are drugs like ketamine, PCP, nitrous oxide, and xenon. Other than xenon, the other place that they're found in nature is you can get nitrous oxide in a particular bacteria, apparently. Just learned that too. And then you have your atypical dissociatives. These are kappa opioid agonists, such as Salvinorin A, which I'll talk to you about a bit, which is found in Salvia divinorum. But there's other kappa opioid agonists that have actually a longer duration of action, which I hope that one day we'll be studying them. So for example, like anadeline, which has been administered in humans, and there are a couple studies that exist, but it hasn't been very deeply studied. Also, ibogaine has some affinity for the kappa opioid receptor. And then you have your deliriants. These are muscarinic antagonists, such as scopolamine and atropine, that are found in belladonna and datura. There's also myristicin, which seems to be a deliriant, and it's found in nutmeg. And then lastly, I always like to bring this one up because I think people always forget about these, the hypnotic dissociatives. So they bind to GABA A receptors, and unlike GABA A positive allosteric modulators, the sedatives such as alcohol, Ambien, and benzodiazepines, these bind directly to where GABA binds. And it's funny because people always include this mushroom in their slides, but this actually contains the hypnotic dissociative muscimol. And I hope again one day that this will also be studied. Other than hypnotic dissociatives, all of these drugs have been, the class of these drugs anyways, have been explored for their therapeutic benefits, such as for depression or anxiety. And all of these drugs, other than the dissociative anesthetics, have also, they've been used for kind of shamanic purposes, ritualistic purposes by various native groups. So this makes me, ask the question then, are classic psychedelics special? That is there something about these drugs that are, let's say, more therapeutic, or can maybe teach us something about cognition more so than these other drugs? Or is there a reason why that we have all these centers that are popping up that are really being named after this specific class of drugs, the serotonin 2A agonists? Or is it just the case that, these drugs happen to be more fun and all the funders have done them and they're just giving us a bunch of money to do research with these drugs? So I'm not going to be able to answer these questions, but hopefully I'll give you some pieces of information to think about, and you can answer this for yourself. So here's the outline of my talk. So in the first part, I'll talk about the acute effects of hallucinogens on functional connectivity, and I'll go into the study we did with Salvinorin A. Then I'll talk about the acute effects of classic psychedelics, as well as various other drugs on cognition and specifically episodic memory. And then finally, I'll end with this study that will hopefully be published soon, in which we look at the what happens after the trip is over. So the enduring effects of classic psychedelics, in this case, psilocybin, on brain function and cognition in patients with major depressive disorder. So I've been told that I could talk for about an hour, and then there's about 30-ish minutes of questions. Basically, I have till, I guess that would be 7 o'clock there, it'd be 10 o'clock here. And so after each part, I'm gonna open it up for some questions. And please ask questions, because if you have a question, chances are somebody else also didn't understand what I just said. And I realize this is supposed to be meant for a medical audience, maybe not necessarily a totally lay audience, but not necessarily a neuroscientist. And I'll be honest, I didn't totally change up my slides too much, but I did add more examples, and I'm going to spend more time trying to explain some of these concepts. So please do ask questions if you have any. So let's just jump right into the acute effects of hallucinogens on functional connectivity. So just so that we're all on the same page, functional connectivity, or static functional connectivity, is the strength of association between the activity of two regions over time. So to make that concrete for you all, here's the default mode network, and here's activity from a few nodes in the default mode network. And you can see that, for example, the posterior cingulate cortex, the PCC, which is that red line, and then the medial prefrontal cortex. These are two of the major hubs of the default mode network. The MPFC is going to be the green line. You see that their activity kind of tracks each other. And so this would then suggest that these two regions of the brain are communicating. It might be that they're passing information back and forth, or it might be the case that, for example, one region is influencing the activity of the other region. And so these regions form these networks, and it's been shown that classic psychedelics can decrease default mode network functional connectivity. This is during resting state or task-free conditions. And so this has kind of become the basis for the entropic brain hypothesis / rebus, because functional connectivity of the default mode network has been, default mode network has been associated with process like self-referential processing. And so it's also been proposed to be the top of a hierarchy in the brain. And so if you destroy what's at the top of the hierarchy, then all this chaos and entropy might ensue from lower levels. And this is what might produce ego dissolution. This is a reverse inference that people make. They're saying that, okay, this is associated with self-referential processing, even though it's associated with many other things, including the opposite of self-referential processing, which is processing what other people are thinking, which is theory of mind. But the idea is that because it's involved in self-referential processing and you now remove activity in this region, or you disrupt it in some way, that this must be related to ego dissolution. However, only one study, I think, as far as I know, has actually found a correlation with ego dissolution, and ego dissolution has correlated with various other things that have nothing to do with the default mode network. It's also been shown that you get larger decreases outside of the default mode network, including from the people who propose that the default mode network seems to be kind of the most important mechanism of psychedelic drug action. And then, as was being said earlier, that all these other drugs do impact the default mode network, including drugs that might make people more egocentric. So for example, like alcohol and amphetamine. So I'm not totally convinced that the default mode network is as important as everyone says it is. Oops, there we go. And so it's also been shown that networks that don't normally communicate as much together at rest, that you can get increases in functional connectivity between these networks. So for example, it's been shown with ketamine, LSD, and psilocybin that you can get increases in the connectivity between the salience network and the default mode network. However, there was a study that came out that looked at 3 different sets of, they had their own data, LSD data, and they then compared this to psilocybin and LSD data. And they never found the same between network increases across all 3 studies. So if I have 8 different networks in the brain, that'll give me 28 different between network pairings. And never was there one single between network pairing that had all 3 studies show the same effect. So I think this is in part due to the fact that we're not constraining cognition. And I have more to say about this, but basically non-specific cognitive operations are going to result in different things happening across every time you scan somebody or a group of people. And then the average of that will show some between network increases that might not actually be all that telling of what's going on in the brain. So if you can't tell, I'm not really the biggest fan of task-free functional connectivity, even though I'm about to go over a study in which I do just that. But before I do, before I tell you about what we did with Salvinorin A, I have a video I want to show you all. For those of you who don't know what Salvinorin A is, because this is an age-restricted video, I have to share screens with the actual YouTube link here. Here it is. Did that work? There it is. Okay, so everybody can probably see this. All right.
Hello, and welcome to Driving on Salvia. I'm Eric, your host. Now, today we're going to take the car out into the streets, and we're going to do some K-turns, and then we're going to do some U-turns, and then we're going to go ahead and parallel park in a spot smaller than we have before. To start it all off, however, we need to take a hit of our Salvia. Once you have that done, you're going to hold it in for about 20 seconds or until your vision begins to vibrate. At that point, you can start your car and you'll be good to go. In the meanwhile, check your mirrors. Make sure that they're pointing in the right direction. I'll be back in a moment.
So as you can see, the onset is pretty rapid.
Oh, I waited a long time. on that one.
You ended up waiting about 40 seconds, which is what we actually do in the lab.
Normally I'm supposed to hold you. Salvia, so long. Otherwise, it makes you trip into space. Excuse me. I have to go to space now.
So you can see he's already getting like visuals, hallucinations. Salvia can be kind of scary. Standard effect of salvia seeing cats. Just kidding. No one saw cats in our study. So I'm going to speed up a little bit. He's kind of confused, but trying to figure out what's going on. And then about right here, he might start clicking back in and remembering what he was doing. He found it pretty fun. Okay. Exit that. Okay, great. So let me share my screen again. So yeah, that's salvia. So Salvinorin A is the main psychoactive constituent found in the plant Salvia divinorum. In various states, salvia is still not regulated. So for example, in Maryland, it's not, and I think in California, it's also not. It's a kappa opioid receptor agonist, and to my knowledge, there had never been a study that had used fMRI to look at the effects of a kappa opioid agonist. However, there was a study with EEG that found various frequencies were kind of desynchronized in the brain, and that this is actually another effect that's been shown with ketamine and psilocybin and LSD, various psychedelics. So in our study, we used a single blind, placebo-controlled within subject design. We used 15 micrograms per kilogram of vaporized Salvinorin A, which should tell you something about the potency of this drug. We only had 12 subjects. It's a bit of a pilot study, but I used some relatively conservative statistics, so hopefully you believe me. And these were all male subjects, unfortunately. They were just the first people that were eligible for the study that could come in. And these were all young to middle-aged hallucinogen experienced healthy adults. So they came in for 2 sessions. In the first session, we just had them practice the inhalation procedure. This is also when we made sure that they could handle the drug and not move around too much while they laid on a couch, because it's bad if you move inside of a scanner. It was also when we collected their subjective drug strength ratings. So we had people on a scale of 0 to 10 rate how strong the drug effects were every minute for the first 5 minutes, and then every 5 minutes after that. And then, oh, I see that I'm a host. Okay, great. Thanks. Somebody else took care of that. And then the next day they would come in, and then this is when we actually scanned their brains. And so there was always a placebo scan followed where we just administered hot air, followed by a Salvinorin A scan where we administered Salvinorin A, although we told people that they could receive Salvinorin A for either one of those scans to try to minimize expectancy effects. While they're in the scanner, there was no task. They just wore eye shades and listened to an ambient track. So you're probably wondering how we managed to get Salvinorin A into the scanner. So that's me holding this bulb of Salvinorin A. The little white blip is the Salvinorin A, which again tells you something about the potency of this drug. And then on the right over there, that's that bulb now attached to a vacuum adapter, which is then attached to a tube that leads into the scanner room. And from the outside, I would then blowtorch what's essentially our FDA-approved crack pipe. So here's the first thing I'll just show you is the subjective drug strength that we got on the day before that they came to the scanner. And what you can see is similar to what you saw in that video. In the first minute or two, people are getting peak effects, and then there's a steep drop. And then afterwards, there's quite a bit of variability. That is, some people are reporting that they're dead sober after 5 minutes, whereas other people are saying that they're getting lingering effects that persist for 10, 20, even 30 minutes onward. So the next thing I decided to do, given that there's all this hype about the default mode network, was look at what happens to the default mode network. So given that the effects of Salvinorin A are super rapid, I split my scans in half to look at what happens during the first scan compared to what happened to the second scan. And I think many of you are probably wondering, why didn't you split your scans into smaller bits, like 5-minute bins? To get accurate estimates of functional connectivity, you usually need 10 to 15 minutes of resting state. However, I did split things into 4-minute bins at one point. It didn't really change the story, but it was a lot noisier. So on the x-axis, we have time. On the y-axis, we have average functional connectivity across the whole default mode network. And then in lavender, we have what's going on in the placebo condition. And then in fuchsia, this is what's happening under Salvinorin A. Each one of those paired points is a single subject. And so what you can see is that every single subject showed a decrease in default mode network connectivity in that first 10 minutes. But if you look at the second 10 minutes, things are a little bit all over the place. That is, some subjects, it's looking like their default mode network remained less functionally connected across that whole scan, whereas other subjects, it's looking like their default mode network might have even have been rebounding. So what I wanted to do was see whether or not this variability in terms of the duration of Salvinorin A on the default mode network was related at all to the variability that we saw in terms of subjective drug strength. So what I did was I subtracted the second 10 minutes minus the first 10 minutes to generate of the Salvinorin A scans default mode network functional connectivity, with the idea there being that a larger change in functional connectivity would reflect those subjects whose default mode network was coming back online. And then I correlated that with the area underneath the curve from that subjective drug strength rating curve for the first 20 minutes, with the idea there being that a larger area underneath the curve would reflect a longer duration of subjective drug strength. And sure enough, we got this negative correlation. That is, those subjects whose default mode network remained less functionally connected across the whole 20-minute scan were also those subjects who were reporting a longer duration of subjective drug strength. So you're probably wondering, well, what about the rest of the brain? So what I'm showing you here are matrices of t-values, comparing the first half of the Salvinorin A scan to the first half of the placebo scan on the left, and then the second half of the Salvinorin A scan compared to the second half of the placebo scan on the right. What you can see is there's less changes. What I should also mention is that anything in fuchsia is an increase in functional connectivity. Anything in green is a decrease in functional connectivity under Salvinorin A. And on the diagonal, we have within network connectivity. So that green square with a star in it, that's that decrease in default mode network connectivity that I just showed you. And then anything off the diagonal is between network connectivity. So for example, that square now that I'm highlighting would be the medial frontal network's connectivity with the default mode network. Now, when I do a correction for multiple comparisons, the only thing that survives is that decrease in default mode network connectivity. So unlike what some people are making a big deal of the default mode network about for the effects of psychedelics, we actually found that the strongest effects of Salvinorin A really were within the default mode network. However, it's worth mentioning that if you look at that diagonal, most of those are numerical decreases in functional connectivity. And if you look off the diagonal, most of those are increases in functional connectivity. So between network increases. So just like with classic psychedelics and with ketamine, we're getting trends towards decreases within network connectivity and increases between network connectivity. Now, another way I can look at within network connectivity is looking at the strongest functional connections in the brain. That's how we a lot of times define networks, or what are the connections in the brain, or what are the regions of the brain that are communicating a lot with each other. So the way I did this was looking at which set of connections in the brain can survive a Bonferroni correction for all 35,778 different functional connections. And I get maps that look like this. What you end up getting are these bilateral connections between homologous regions. So usually two regions in the brain on either side are going to be in the same network. And so a functional connection between them would then count as a within network connection. And what you can see is that most of these within network connections tend to decrease under the effects of Salvinorin A. So again, this is just another way for me to show you how within network connectivity is decreasing in the brain. So what I just showed you was static functional connectivity. So how things are connected on average across a whole time series, or in this case, across 10 minutes. But what might be the case is two regions of the brain might be in general pretty much, talking to each other a lot, but they might go in and out of sync. And this is not something that you could capture usually with a single measure of functional connectivity. So what some people do is they'll take a window of their data. And so you can now take, in this case, I took 5 frames, and now you can look at functional connectivity of that window, and then you can slide that window down to generate a time series of functional connectivity values, or Pearson's correlations in this case. Now, this is not exactly what I did. I actually used a more complicated method known as dynamic conditional correlations. It gives you far more reliable data, but the idea is the same, that we're trying to estimate this time series of correlations. And with the power of those correlations combined, I can generate a variance term, which I'll now refer to as dynamic functional connectivity. But given that there's this idea of entropy being important in the brain, I can also take the entropy of that time series to, I'm going to call this entropic functional connectivity. So variance and entropy are both measures of dispersion in your data. And at their lower bounds, they do tend to be associated with each other. So zero entropy would also be zero variance, but they don't necessarily have to be related. So just to make this example concrete, if I were to draw 10 integers at random between 1 and 10, and I wanted to get the largest variance, then I would draw 5 1s and 5 10s. So the idea with variance is how different are your values from the mean. So the mean in that case would be 5.5, and I got the values that are most dissimilar from 5.5. And so that would be, example of a high variance. But to get high entropy, what you would want is how close is your distribution to being uniform. So for example, in that example I just gave you, what I want to do is draw one of each number. So 1, 1, 1, 2, 1, 3, etc. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That would give me a uniform distribution, and that would be the highest amount of entropy I could produce. Another way of thinking about entropy is, for example, something that's like fully entropic would be like a fair coin, so that half the time it's flipping heads, the other time it's flipping tails. So that's a uniform distribution. But if you had an unfair coin, this would now be less entropic because it's more predictable which state you're going to go into. So not the exact same things. So the first thing I'll show you is what happened to dynamic functional connectivity, and what you can see. So again, there's these T matrices, the diagonals within network connectivity, off the diagonals between network connectivity. On the left is the first half of the Salvinorin A scan. On the right is the second half. And what you can see is that just in general across the whole brain, dynamic functional connectivity or variance basically decreases. If you look at what happens with entropic functional connectivity, on the other hand, nothing really survived a correction for multiple comparisons, but it's kind of looking like entropy increased. So one way to kind of think of these data is that although maybe the brain is becoming more similar under the effects of Salvinorin A from time point to time point, that there's far more states in there, even though those states might be more similar to each other. But this is, just massive speculation, and it's a bit reverse inferential. So the last thing I'm going to do is I'm not going to go into the details here. You can read the paper, but I used a classifier. I trained a classifier on a subset of my data to then predict the other subset of my data. And what I ended up doing was I trained it on either just on within network connectivity or just certain between network connectivity. And what you're seeing here is performance of that classifier. So what I'm showing you is how well that classifier is able to correctly identify a scan as being Salvinorin A, while correcting for how often it's mislabeling something as not Salvinorin A. So how often it's mislabeling the placebo scans and calling those Salvinorin A. So what you're looking at are discrimination scores. And so I like to call these Trump matrices, because with greater color comes more discrimination. And so what you're seeing on the left, what you're seeing in the center is that dynamic functional connectivity ends up being the best predictor of whether or not the brain is under Salvinorin A, kind of regardless of which network interactions you're looking at. But it's worth mentioning that the default mode network again pops out for both static and entropic functional connectivity. So just to conclude real quick, we found robust reductions in functional connectivity of the default mode network, especially in that first 10 minutes. And this was related to subjective drug strength. We also found decreases in within, but increases in between network connectivity, just like what's been shown with psychedelics and ketamine. We found whole brain decreases in dynamic, but increases in entropic functional connectivity. And using this Connectome-based classification method, I found that dynamic functional connectivity was the best predictor of whether the brain was under the influence of Salvinorin A, but it's worth mentioning that there were some motion artifacts that we have like 30 pages of supplemental material in the paper that we try to account for. And even though, by correcting for some of those motion artifacts, we actually got better statistics, it's still worth mentioning that you never fully remove those artifacts. And so all of this should be taken with a grain of salt. And this is actually something that's true of all psychedelic studies that people a lot of times fail to mention. And so just one last part is that it did turn out that default mode network functional connectivity and the entropic functional connectivity of it were also good predictors. So this whole idea of, rebis being especially, particular to, for example, serotonin 2A psychedelics does not seem to, hold up. As I mentioned, the default mode network is being, it gets hit by various drugs, including non-hallucinogenic drugs, and that they don't even find the strongest effects of psychedelics on the default mode network. Same thing with entropy. Entropy is larger increases in entropy are found even in sensory networks, which would be considered lower level. So this is one study where it's only one study, but we did find that the most selective effects were in the default mode network. So before I move on to the next part of my talk, are there any questions?
I think everybody understood that perfectly, 100% of that perfectly. Yeah.
Okay,
You can do a lot of questions at the end. I mean, I would love to hear a translation of an even dumbed down version of that at some point. But I do, appreciate the conclusion slide.
Okay, cool. Well, I see that in the chat, there's still no questions. So I will just keep going on. So, the problem with, what I was just mentioning there is we're looking at resting state, and you can't really infer what's going on in the mind very well with resting state connectivity without having any behavior. This has been kind of a problem I see in a lot of psychedelic work, is that we're just kind of ignoring experimental psychology, whether that means using task-based fMRI or just pure behavior. And so, what I'm going to now go over is, what do classic psychedelics do to cognition? And so the first thing, a lot of people want to say that that psychedelics enhance cognition, and there have been a few cases in which they acutely do so. So there was a study in the 90s that found psilocybin increased semantic priming. And then there's a drug, MDEA. It's a lot like MDMA, except it's an ethylamphetamine instead of methamphetamine. And in this particular study, they actually took the R enantiomer and the S enantiomer separately. So what a lot of people don't realize about MDMA is that really in most human studies, or nearly all current human studies, really you're administering two drugs. The S enantiomer is what gives you the stimulant effects and the lovey-dovey whatever effects, because they increase synaptic serotonin, dopamine, and norepinephrine. Whereas the R enantiomer doesn't really do that much. And if anything, it binds to the serotonin 2A receptor, giving MDMA weak psychedelic effects. So in this particular study, they separated the two enantiomers, and they gave just the R or just the S enantiomer. And the R enantiomer, sure enough, did produce psychedelic effects, whereas the S enantiomer produced more of the kind of standard MDMA-like effects. But the R enantiomer also did something else, where it enhanced the pop-out effect. So this is being able to catch a stimulus in an array that is different from all the other stimuli. So this will usually pop out at you, and it popped out even better under the effects of R-MDEA. And then there were some studies looking at divergent thinking with ayahuasca, and they found that in these studies, divergent thinking increased. However, these studies were done such that people performed the task before they received ayahuasca, and then they performed the task after they received ayahuasca. So any improvements in performance could simply be due to practice effects. That is, if you do a task twice, you'll probably do better on it a second time. So not really a lot of great examples of how cognition can be enhanced acutely under the effects of psychedelic drugs. However, if you look at how they can be impaired, it's pretty clear that psychedelics do impair cognition. And for whatever reason, a lot of people don't want to believe this. So for example, they'll say, well, okay, motor action is largely preserved under the effects of psychedelics. And what you can see there is they have no citation for that. What they do cite is that they say there's less serotonin 2A receptor expression in motor cortex. But what's worth mentioning is that there's actually pretty high expression of serotonin 2A in motor cortex. Although less so than association cortices, it's still higher than many regions in the brain. And this whole statement is just wrong, because it's actually been shown that psilocybin does impair psychomotor function. So then they'll say, okay, well, that's, conventional cognition is impaired, but unconventional cognition or something is not impaired. In this case, they talk about cognitive flexibility. And the way they justify that cognitive flexibility might be enhanced is that if you give a serotonin 2A antagonist, you block the receptor, that impairs cognitive flexibility in animals. So then the inference would be that if you give an agonist and that activates the receptor, that should then enhance cognitive flexibility. They do say, well, we'd be hesitant to infer from this that psychedelics can enhance cognition. Well, hesitate no more, because it was actually shown recently that LSD does in fact impair cognitive flexibility. This was also shown in animals recently as well. So then they'll then say, well, this all just might be due to generalized disengagement. Now, this is something that really bothered me, because it's basically just writing off the whole field of experimental psychology as being useful in psychedelic science. Not only if any of you have done any experimental psychology, you know that your undergrads aren't really engaged in the first place. And then second, if any of you have done any drug studies, you should know that under any drug, people disengage from the task. So this is an example of what I've been referring to. I've kind of coined this term as psychedelic myopia or psychedelic exceptionalism, where people think that psychedelics, for example, are kind of outside the realm of other aspects of psychology or neuroscience. There's something special about them that makes them such that you can't study them like other drugs or other aspects of cognition. Also, this whole idea of generalized disengagement cannot explain this pattern of effects. So what was shown recently by Fred Barrett, my boss, was that in a study, they gave either dextromethorphan or psilocybin, and they showed that under psilocybin, you got these massive impairments in working memory. I realize if there's any of my working memory friends here, I'm sorry, I'm referring to the NBAC as working memory, and I realize that's not the best measure of working memory, but at least in this task, on this particular task, psilocybin massively impaired performance on the NBAC, whereas dextromethorphan had little to no effect. In contrast, in the episodic memory task, dextromethorphan massively impaired episodic memory encoding, whereas psilocybin didn't have very large effects. Generalized disengagement should predict that psilocybin should be worse across the board than dextromethorphan, but this double association tells you otherwise. Also, I found with my MDMA study, as I'll talk to you about in a little bit, MDMA acts like a psychedelic in terms of its effects on episodic memory. I found that MDMA it impaired emotional memory more selectively than neutral memory. In fact, I think the neutral memory effects weren't even significant. So again, generalized disengagement should predict that you get equal impairments across all types of stimuli, and that was not what we found. So I just, really want to put that point out there that you can't just write off a whole field and, just have this cop out that generalized disengagement. This is psychedelic exceptionalism or psychedelic myopia that we need to stop thinking like that. So before I get into episodic, what we did, I have to define a few terms. So first of all, what is episodic memory? So this is to be differentiated from other types of memory, such as habit learning or skill learning or conditioning. This is the conscious re-experiencing of information from the past or mental time travel, in the words of Wendell Tolving. And when we're talking about, so for example, this is thinking about, what you maybe ate for dinner earlier, like an hour ago or something. And so when we are talking about targeting or manipulating episodic memory with drugs, we really have to be careful of which phase of memory that we're trying to target. So you have the encoding phase in which you're studying or learning information for the first time. You have this consolidation phase in which after that, these memories now need to stabilize. And so that if you administer a drug, let's say after encoding, this can also now interfere or perhaps even enhance the stabilization of these memories. And then finally, when you're testing your memory, this is known as the retrieval phase. And this is when we have you actually try to remember what you saw. And just to complicate things further, we have different processes that we like to think about in episodic memory. So recollection is kind of the more intuitive form of episodic memory. This is your retrieval of specific details and associations, such as where and when information that's supported by the hippocampus. In contrast, familiarity is the less intuitive form of memory. This is more just an ambiguous feeling of knowing that something's happened without any kind of corroborating evidence. So because this is less intuitive, the example I'll give you is one day you guys might see me at a conference and you might say, oh my God, you look so familiar. I can't remember where I met you, when I met you, what your name is. I just know that I've met you. That would be an example of having high familiarity in the absence of any type of recollection. Something about my face was familiar. And then lastly, there's meta-memory. So this is a form of metacognition. This is how well you understand your own memory abilities or your own memory system. So there's this idea that psychedelics are mind revealing. The term means mind manifesting or mind revealing. So there's this idea that they might reveal the contents of one's mind to the individual, or maybe perhaps the underlying mechanisms, right? And so you might think that metacognition might be enhanced under the effects of a psychedelic drug. So we decided to look at all these different aspects of memory. But before I show you exactly some of the results, I'm going to give you, because a lot of you are probably wondering, how do we test memory in the lab? I'm going to give you a little example that involves pictures of my late cat. So during the encoding phase, I'll show you a bunch of pictures and I'll have you make some judgment about that picture, some judgment that I don't really care about, but it's just so that you engage in the task. So for example, over here, I'm going to ask you, does this picture contain a human? So in this picture of my cat really high on catnip, you should say no. Then, in this picture of my cat passed out on someone else who's passed out, you should say yes. And then in this picture of my cat, that's not a human, that's a rat, so you should say no again. So then usually there's some kind of delay. This is when a lot of times we like to wait for drug effects to dissipate. So if we're trying to target the encoding phase, I would have given you a drug before that task, then you would have performed that encoding task, and then we would wait some time, perhaps even a day or two, so we can make sure that the drug effects don't impact memory retrieval. In the cannabis literature, this was the problem until recently, where they assumed that there was two different effects that were happening in cannabis, and those were all due to the encoding effects. It turns out one of those effects was due to memory retrieval when somebody did the proper study. So that's somebody being me. And so now here's the memory test. So here's the retrieval phase. So in addition to just asking you, do you remember seeing this picture? I'm also going to ask you to rate your confidence on this 6-point scale. So 1 would be that you're certain that it's new, and 6 would be that you're certain that it's old. And we ask you to use the full range of responses. So over here, you should probably use one of the last 3 buttons on the right, because this was in a picture that you previously saw. Here's a picture with my cat with a rabbit. That's not a picture that I previously showed you, in which case you should probably use one of the 3 buttons on the left. Here's a picture of my cat high on catnip. Again, you should probably say this is old. Here's a picture of my cat with that rat. You should say it's old. Here's my cat burning down the world. You should say that's new. And then here's my cat going to the cafe. You should say that it's new again. So memory confidence, the reason why we care about memory confidence is because it can reflect multiple things. One, it can reflect like the strength of a memory, so maybe how vivid you remember something. It can also reflect the amount of information. So perhaps from that picture of my cat passing on my roommate, you might remember, oh yeah, the guy had a white shirt on. He was kind of had like Nicolas Cage type hair, and my cat's like, left arm was all over him. In which case you had all these kind of details that can also influence your confidence. And then finally, you might also have high confidence because you have a good understanding of your memory system. So, your confidence can also reflect how well you know what you think you know. This is why it's called meta memory. And so it's possible to have a bad memory and also have a bad meta memory. I think we all know a lot of older adults that have poor memories and don't want to admit it. Or we have individuals like myself, in contrast, who have a poor memory, but because of studying memory for so many years, for over a decade now, I understand that I have a bad memory. And so hopefully I probably have good meta memory. And so using these different signal detection models, I can estimate these different parameters: recollection, familiarity, and meta memory. I don't have time to go into them into detail, but I'll be happy to answer any questions about them later. So what I'm going to do is I use this bootstrapping procedure to generate distributions of parameter estimates for recollection, familiarity, and meta memory that end up looking like this. And I'm going to show you what happens with a bunch of other drugs to try to contextualize why what I think is happening with psychedelics might be special. So anytime you see a gray distribution, that's going to be the placebo condition. The top will always be recollection, the middle will be familiarity, the bottom will be meta memory. The blue distribution will always be when a drug was administered at encoding, and that will be the thing I'm going to be focusing on here. But there'll be a few times in which I will mention the other phases in which drugs hit, so consolidation or retrieval. So anything to the left of that gray distribution will be an impairment. Anything to the right of that gray distribution will be an enhancement. And what you can see here is that alcohol, when it's administered at encoding, right before people are studying a bunch of information, you can see that it massively impairs recollection, it impairs familiarity, it even impairs meta memory a little bit. And the reason I decided to bring this in is because this has nothing to do with psychedelics, but I think this is one of the coolest effects in all of psychopharmacology, cooler than anything I've seen so far with psychedelic drugs. And so I have to bring this up. If you notice that if you administer alcohol immediately post-encoding during the consolidation phase, you actually enhance memory. And so you can see that in that top panel for recollection. This is known as retrograde facilitation. It's one of the most paradoxical effects because obviously if you study when you're drunk, you're not going to remember anything the next day for your test. But what this is saying, if you study and then get drunk immediately afterwards, you might actually remember more for your test than had you not drank at all. And now I don't want to say people should go get drunk after they study, but what I will say is that retrograde facilitation has been shown since the 80s across multiple labs around the world. It's been shown with multiple different types of stimuli, including words, sentences, pictures, emotional pictures. It's been shown after multiple delays, so testing people immediately after you give them the drug, after a nap, 1 day later, 48 hours later, etc. And it's also been shown after other GABA-A positive allosteric modulators. So it's been shown with Ambien, benzodiazepines, and alcohol. So do what you will with that information. But again, I think that retrograde facilitation is one of the coolest things that's ever been shown in psychopharmacology. So here's another sedative. Here's a benzodiazepine triazolam, 2 different data sets that I reanalyzed. The light blue would be the higher dose. And you can see that triazolam impairs, when it's administered at encoding, impairs recollection, impairs familiarity, not really much of an impact on meta memory. So now we get into, is this not, there we go. We get into THC. This is a crowd favorite probably from some of the people in California. You get an impairment in recollection, an impairment of familiarity, and oddly enough, we found this enhancement of meta memory. So if anybody, I'd be surprised if this is replicated. If anybody has any THC data in which THC was administered prior to encoding and that people gave confidence judgments during retrieval, I would love to reanalyze those. But perhaps this makes a little bit of sense. Some people, a lot of people are overconfident in their memory. They think they know more than they actually do. And so maybe if they were on THC, they might think that, I don't remember anything from that day I was high. And they would be correct to think that. Because as I showed you, they were impaired with recollection familiarity, and in which case now their confidence is better tracking their accuracy. So now we're going to get into some of the hallucinogens. So here's 2 different data sets with dextromethorphan. And again, in light blue, we have the highest dose, which I think in this study was 300 to 400 milligrams. And you can see that you get impairment in recollection, a impairment of familiarity at the higher doses, and what looks like sometimes, at least in one of these data sets, an enhancement of meta memory, but it wasn't consistent. Now here's ketamine, another dissociative, impairment in recollection. You start to see an impairment of familiarity at the higher dose. And weirdly enough, you also again start to see this little boost in meta memory, which might or might not be real. But now let's move on to what happens to drugs that enhance cognition. So for example, this would be stimulants. So here's a study with dextroamphetamine on the left, and on the right, we have a study with methamphetamine. And so what you can see is when amphetamine was given at encoding, that would be the blue distributions, you get really not much of an impact on recollection or familiarity, but you do seem to get this enhancement of meta memory. What was really cool in this study is that dextroamphetamine was also administered at retrieval and showed that meta memory enhancement, and it was also administered at both encoding and retrieval. That is 2 separate sessions, and you get a really large enhancement of meta memory. So this is the first instance. So although people have shown that stimulants can enhance cognition, I think this might be the first instance in which a stimulant's been shown to enhance metacognition. So what about a drug that has stimulant-like effects, but also has psychedelic-like effects? That would be MDMA. And so here's a study with MDMA that was administered at encoding or retrieval. Let's just pay attention to the blue distribution. You can see that there's an impairment in recollection, oddly enough, an enhancement of familiarity, and not really much of an impact on meta memory. So in this case, MDMA is really acting nothing like the stimulants in terms of its effects on episodic memory. So is this due to the fact that the R enantiomer binds to the serotonin 2A receptor, or is it due to something else? MDMA has other pharmacological differences from amphetamine and methamphetamine. Well, one way to test this would be to look at what happens with a more selective serotonin 2A agonist. So that would be the study with psilocybin. And sure enough, with psilocybin, we find that there's an impairment in recollection. It starts to get an enhancement of familiarity when you get these larger doses at which people are barely even functional, and then no impact on meta memory. So what's kind of interesting about this study was that all these drugs that impair recollection at their higher doses impair familiarity, except psychedelics. With psychedelics, we get this impairment in recollection. You start to see a sparing or even enhancement of familiarity at those higher doses. And now there's other conditions in which where recollection is down and familiarity is up. That is when these 2 processes can decouple. And people have shown this in the lab without drugs. So there's situations in which you can evoke, when you can reduce recollection but increase familiarity. And people will report feeling deja vu, feeling like they're having an insight when they're not actually having insight necessarily, or feeling like they can predict the future. So if that doesn't sound like somebody on psychedelics to you, I don't know what does. And as you can see, we didn't really see any impact on meta memory. This idea of psychedelics being mind revealing was not supported here. But this was just one aspect of metacognition and really only one aspect of meta memory. We had people assess their confidence at retrieval. And so we're looking at how well their confidence, their memory during retrieval aligns with what they previously saw with their actual memory performance. Instead, we could look at prospective meta memory. So this is during the encoding phase. We would ask people, how likely is this that you're going to remember this later? So perhaps I see a picture of a tiger and I say, oh, I'll definitely remember that picture. See a picture of a can, I might say, I probably won't remember that. And so how well your confidence aligns there would be a measure of prospective meta memory. And perhaps in that situation, psychedelics might enhance meta memory, prospective meta memory. You might think that, for example, people having a mystical experience, something very meaningful, and that they might forget everything else around them at that moment, but they know what they know in that very moment. But this remains to be tested, and actually it's something that we're working on soon. So before I go to the last part of my talk, are there any questions over what I just went over? See that there's the chat. I feel like let's see. I do see one question in here.
There's a couple from earlier. Yeah, Alec had one, a good one back in talking about cognitive flexibility and psychological flexibility and the mismatch between them, potential mismatch between them.
Well, that's interesting. Well, I wish you were one of my reviewers, because somebody asked us to talk about psychological flexibility with cognitive flexibility in the study that I'm about to go over. So yeah, I don't know if those 2 things are necessarily fully related to each other, but they do kind of sound like it on paper. And maybe I should go over the study, and then that question might make more sense, this next study. But while we see people, I do see talks, people asking specifically about this study. Okay, so there's a non-specific question about genes. If okay, so I see one now. Is retrograde facilitation observed with any other pharmacological classes other than GABA A agonists? So there was a review on this by John Wickstead in which he was predicting that NMDA antagonists should also show retrograde facilitation. And the reason for this is because at the synaptic level, so there's a couple really, there's two explanations for retrograde facilitation. There's a really boring one, and then there's maybe a slightly more interesting one. The boring one is that if I'm learning new information after I learn old information, that new information is going to interfere with that old information. So if I get drunk immediately after studying something, there's no new information coming in that's going to interfere with the old information. That's why you get a better memory. So if that were true, then this would be true of all other amnestic agents, including THC or GHB. Those studies have actually been done, and you did not see evidence of retrograde facilitation. So what somebody has also done, this is Sarah Medic, who's at UC Irvine. What she did was she had people take a nap immediately after studying stimuli. And then she either gave people Ambien, GHB, or a placebo. So first of all, people are going to sleep. And so there's very little interference there's very little new learning that might interfere with old learning. And then afterwards, she then tested people's memory, and only Ambien worked in that situation. And what she also did was she looked at people's, she had EEG going on during their sleep. And she showed that what Ambien was able to do was increase the power of slow wave oscillations. It increased, I think, sleep spindle density, and it also increased the coupling of the sleep spindles to the up state of the of slow oscillations. So all those are kind of signals of memory stabilization or consolidation. And so basically, I think that there's actually an active process that's being facilitated when retrograde facilitation happens. From sedatives that might not actually happen with NMDA antagonists, but some people predicted that NMDA antagonists should do that as well. And there's at least one study in which they gave people nitrous oxide immediately after watching a horrific video, and people reported having less memory intrusions throughout their week. So in that case, that would actually be no evidence for retrograde facilitation. There's also a study that was done with methamphetamine that was done from my old lab. Actually, that was those data that I just showed you. Those were that was the encoding data. There was also some consolidation data. And in that particular study, they gave meth because it absorbs very quickly. They also crushed it up and put it into liquid, so that way it would really absorb quickly. And in that study, they didn't show any enhancement or impairment of meth delivered immediately post-encoding. So as far as I know, it really just seems to work with GABA A specifically drugs, and not GABA B, or else there would have been that effect with GHB. So let me see. I see another question here. Also, feel free to shout these out at me, but did you conduct any studies on the recollection, familiarity, meta-memory when subjects were dosed during consolidation? Oh, similar to the ethanol. Well, I just told you that. So yeah, there was a meth study, and that was the only one that's been done. And so, these other effects I was just telling you, THC didn't work, GHB didn't work. They only looked at basically a basic form of memory. They didn't look at meta-memory, for example. So that might be the case that something else might happen there if they look closer at their data. Oh, what's worth mentioning, though, is that in this reanalysis with methamphetamine, so I told you that stimulants at encoding enhance meta-memory, at retrieval they enhance meta-memory. The consolidation data actually found that it impaired meta-memory. So if you give stimulants immediately post-encoding, so if you're going to study, don't take your Adderall afterwards. That might actually impair how well you know that information. So, okay, I said that there were two reasons. So I think those are the two reasons hopefully I went over with retrograde facilitation. Is one that there's a decrease in interference, the other there's active facilitation of stabilization processes. There's potential body preclinical mediated facilitation of thalamocortical oscillations. Okay, great. I see that. I'm gonna now move on to the next part, and then afterwards, the very last part of my talk, and then afterwards we can, I'll just open up the questions to anything that I went over. So here's what happens when the trip is over. How do these drugs affect brain function and cognition? So I should mention that we did this study in a healthy population. I don't have time to go over this, but it's published and you should check it out. So in this study, what we were interested in specifically was, does cognitive flexibility as well as neural flexibility increase after the trip is over in patients with major depressive disorder? And so cognitive flexibility specifically is the ability to adaptively switch between different cognitive operations in response to a changing environment. And this has been shown to be impaired in depression, as well as other disorders such as OCD and addiction that also might be amenable to psychedelic treatment. And the way we measure cognitive flexibility is typically how much people perseverate on an old rule in a task, a rule that's no longer rewarding or no longer correct. So I think the Wisconsin card sorting task is an example that many people know. In this particular task, you get a handful of cards, you get 4 cards or 3 cards, And there's 3 rules. There's 3 different features of each shape, and you have to pick which one was the odd card out based on one of those rules. Once you figure out which rule it is, at some point that rule switches, and then we, to measure cognitive flexibility, or really to measure cognitive inflexibility, is how often you perseverate on a rule that's no longer correct. And so it's been shown that cognitive flexibility is associated with the anterior cingulate can support cognitive flexibility, and that neural flexibility, so basically variability in brain activity, can also support cognitive flexibility. But it's worth mentioning that neural flexibility is not the same thing as cognitive flexibility. Just want to point that out. And then it's been shown that psychedelics, that they tend to especially modulate the ACC, the anterior cingulate, and that they also increase some measures of neural flexibility, such as entropy might be one of those measures. However, it's unclear, this is all acute effects. It's unclear if these effects persist and how long they persist. So there's been some studies looking at what happens one day after psilocybin. And some people will show that you get the same acute effects, although maybe weaker. And then other people are showing you get almost like rebound, like opposite effects. And so what we decided to do was look at what happens 1 week after psilocybin therapy in patients with major depressive disorder. So in this study, we used an open label, weightless control design. So half of our subjects received psilocybin immediately after enrollment or soon after enrollment. The other group had to wait 8 weeks. And the group that waited 8 weeks, we tested, as soon as they were enrolled in the study, we tested their cognitive performance. And then after that wait period, we tested their cognitive performance again before they were ever given psilocybin. So they performed the task twice in order to see whether or not they just improved just based on practice effects. You do a task twice, you're going to get better at it. And we wanted to be able to decouple that from the improvements people might have after psilocybin therapy. So in this study, we gave people 2 doses of psilocybin, a moderately high dose and a really high dose, separated by about 1.5 weeks. And then we scanned people before they ever received psilocybin and 1 week after they received psilocybin. We used a 7 Tesla scanner, which is a high magnetic field, higher than 3 Tesla scanners that are typically used. And although some people might think, oh, hey, more, brain stuff with more, magnetic field. In the wise words of an old advisor of mine at UC Davis, Charn Ranganathan, mo Teslas, mo problems. And that was certainly a problem here. So we actually, with a stronger magnetic field, there's more inhomogeneities, especially around nasal cavities or anywhere where there's like air. And that'll actually produce signal dropout. And so we actually lost, I think, like 75% of the brain in this study. So depending on the subject. So, I wasn't able to do network-based analyses, but I'm going to show you some things that I was able to do with these data. And then we had a resting state scan. We also collected spectroscopy so that I could look at metabolites such as glutamate, because people have shown that different antidepressant treatments can change glutamate levels. And then we measured cognitive flexibility with the Penn conditional exclusion task. This is like the Wisconsin card sorting task, except there's multiple versions of it so that people can repeat the same task multiple times and not get too much better at it. And then we had about 20-ish subjects, depending on the analysis, and these were all young to middle-aged adults with limited hallucinogen use. So the first thing I'll show you is the data. If you saw the JAMA Psychiatry paper that came out, these are those data, except they didn't show individual data points. I'm now showing you individual data points. Anything before that baseline would be the weightless control group. You can see that the weightless control group didn't get better just by interacting with our team and waiting around and expecting to receive psilocybin eventually. They did get massively better after they received psilocybin. And this change was maintained for 4 weeks. And I can also tell you that some of it was maintained up to 3 months, although a lot of people weren't as good after 3 months. And I think a lot of people even went back on their antidepressants. Now, what about cognitive flexibility? As I mentioned, cognitive flexibility is a measure. What we're really measuring is cognitive inflexibility or cognitive rigidity. So less errors on this task is greater cognitive flexibility. Again, look at what happens before the baseline in the weightless control group. People perform this task twice and did not get better at it. However, after they receive psilocybin post-baseline, you get all these decreases in perseverative errors. That is greater cognitive flexibility. So what about what happens with glutamate in the brain? So we looked at the anterior cingulate cortex because we know that seems to be important for cognitive flexibility, as well as for depression, as well as for psychedelic effects. We also looked at the hippocampus because this is a region that's it seems to be implicated in major depressive disorder. What we found was that only in the anterior cingulate did we get decreases in glutamate after psilocybin therapy. This did not show up in the hippocampus, and also N-acetyl aspartate. So I decided to use this region of, I decided to use this area of the ACC as a region of interest. So that way I wasn't just cherry picking whatever gave me significant effects. I literally took what was the region that showed this decrease that overlapped across all of the scans. And then I looked at its functional connectivity with the posterior cingulate. This is the hub of the default mode network. To see what changed with either static or dynamic functional connectivity. Other people have also looked at what happens one day after psychedelic treatment, looking at these two regions. So I decided to do the same thing. And what we see is that static functional connectivity did not change, but dynamic connectivity did. So the brain, so this particular functional connection is becoming more dynamic after psilocybin therapy. And in general, the brain became more dynamic, although this was, one of the significant effects. So then I decided to look at what does this now correlate with the improvements that we saw in cognitive flexibility? And sure enough, it did. What I looked at is the change in dynamic functional connectivity post to pre-scan and looked at how that correlates with the change in cognitive flexibility after 1 week or after 4 weeks. And in both cases, we get a pretty strong or a moderately strong positive correlation. However, if anybody notices something weird here, good for you, because it took me a second the first time I saw this. If you get a decrease in something and an increase in something else, you should expect a negative correlation. That is, if I get a increase in dynamics, that should then predict a decrease in errors on that cognitive flexibility task. Yet we got a positive correlation here. So what this is saying is that those people who had the largest increases in dynamics were actually those that had the least benefit from psilocybin in terms of effects on cognitive flexibility. So this was really weird. And so I decided to, dig deeper into my data. And so this is going to be the most confusing part of my talk, but I'll have a part that, hopefully summarizes this, and this will hopefully be published soon and you can try to dig deeper into it. And so what I did was I looked at, if I look at just baseline brain activity, can I predict baseline cognitive flexibility? And can I also predict changes in cognitive flexibility after psilocybin therapy? The advantage, the one thing that this might be useful for is one day we might finally use fMRI in psychiatry. It's really never been used in psychiatry before. But what this could do for you is, hey, you can look at people's baseline functional connectivity or their brain activity and then say, hey, this person would be a good candidate for psilocybin therapy because it's going to improve their cognitive flexibility or something. So the first thing I decided to do was just look at baseline functional connectivity to predict baseline cognitive flexibility. And so what I'm showing you here, so in this Connectome-based predictive modeling procedure, there's a kind of an arbitrary threshold you can place to allow features into your model, to allow different functional connections into your model. And so what I wanted to see is whether or not I wanted to see a model that was robust regardless of the thresholding that I used. That way you could say, regardless of adding more or less noise, this model still worked. So that's what we have on the x-axis is different thresholds. On the y-axis, we have performance of the model. And then, so how well this model was able to predict cognitive flexibility from baseline cognitive flexibility from baseline functional connectivity. That gray line is a significant prediction. And what you can see is really the only thing that was a significant predictor of cognitive flexibility was baseline dynamic functional connectivity. And in this case, it was dynamic functional connectivity of the full connectome. So I trained it on various different families of models, static, dynamic, or both static and dynamic connectivity, on ACC, PCC, both ACC and PCC, or just every functional connection I had available to me. And basically, if I just gave it a bunch of dynamic functional connections, I was able to predict people's baseline cognitive flexibility. So then I decided to look at what are these features? Which are the functional connections that are getting into this model as I reduce my threshold? And so here's, again, so as I reduce my threshold, that's on the x-axis. On the y-axis is the number of features that were added into the model. And by far, the best predictor was dynamic functional connectivity in terms of predicting from baseline functional connectivity to baseline cognitive flexibility. And what you can see here is that these all of these edges are negatively correlated with cognitive flexibility. So that is, the greater dynamics meant the less errors on cognitive flexibility. So that's what we would expect. That's basically saying that more dynamics means more cognitive flexibility. That's what other people have shown. And that's what we're seeing when we use baseline data to predict baseline data, baseline functional connectivity to predict baseline cognitive flexibility. However, if I now look at what happens if I use baseline data to predict changes in cognitive flexibility, here's the models that I get. Again, both static and, in this case, both static and dynamic functional connectivity were the best performing models. That's what you see in red. And they involved really just the ACC, which this kind of makes sense. Again, the ACC is modulated by psychedelics. It's implicated in depression, and it's implicated also in cognitive flexibility. So static and dynamic functional connectivity of the ACC is predictive of whether or not people will improve in cognitive flexibility. But now I did the same thing that I did, where I now look at those features that are allowed into the model at various thresholds, and I look at what is their relationship to changes in cognitive flexibility. And what you can see here is that, again, dynamic functional connectivity was the best predictor of changes in cognitive flexibility. However, that relationship is now flipped from when we try to predict baseline cognitive flexibility. So in this case, what we're saying is that the greater dynamics at baseline meant less improvements in cognitive flexibility after receiving psilocybin therapy. So just to make this, I realize that this is one of the most confusing things, and it's even hard for me to explain at times. So I'm just going to try to make this concrete. What I think might be happening. So by the way, I was hesitant to include a slide like this because this is not exactly super concrete data, and the next data set that comes out might prove this totally wrong. But here's what I think might be happening. So we have an inverted U. So greater neural flexibility, usually, up to a certain point means greater cognitive flexibility, and then too much neural flexibility, such as, for example, in schizophrenia, might lead to less cognitive flexibility. So you have, for example, somebody who starts over here, and they're not very happy, and then they receive psilocybin therapy, and you get this massive improvement in cognitive flexibility, as you can see with the upward arrow, as their neural flexibility increased. What if you had somebody that started here and then they received psilocybin therapy? Well, now you're pushing them over the edge in terms of that inverted U. You still get some benefits, which most people still showed some benefits in cognitive flexibility, but that benefit is far less. So what now happens if you start here and you receive psilocybin therapy? Well, now you might not be so happy because your cognitive flexibility has now gone down. So this is completely speculative, but this could be what's happening as what we're seeing here. So just to summarize, psilocybin therapy improved depression and cognitive flexibility. We found that decreases in anterior cingulate glutamate. This is actually maybe somewhat consistent with what Draulio and people in Brazil did, where they showed 1 day after ayahuasca that they got decreases in glutamate in the posterior cingulate. And then between the posterior cingulate and anterior cingulate, we got increases in dynamic functional connectivity or neural flexibility. However, greater changes in dynamics were actually associated with less improvements in cognitive flexibility. And then to dig a little bit deeper into my data, I found that baseline static functional connectivity and dynamic functional connectivity of the ACC was predictive of improvements in cognitive flexibility. However, too much baseline dynamic functional connectivity might be detrimental to larger improvements in cognitive flexibility. So just to kind of summarize everything. So, are psychedelics special? It was kind of the question I started with. And so in terms of the effects on task-free functional connectivity, at this point, I'm going to have to say no. There's nothing special about the default mode network. Are they special in terms of the effects on episodic memory encoding? I don't know, maybe so. It seems like it compared to all these other drugs. I just gave you a small snippet of what I'm doing with those data. This is a huge manuscript, and the only drugs that looked like that were psychedelic drugs. So it looks like, psychedelics might impair recollection while sparing or even enhancing familiarity. What about their effects on major depressive disorder? Well, other people have shown with other antidepressant treatments that you can get improvements in cognitive flexibility, and that certain other drugs might also increase neural flexibility. But, I think that this is promising that at least psilocybin might be useful for the treatment of major depressive disorder, as well as maybe other disorders that have impairments in cognitive flexibility. So I just want to say thank you to my lab. Thank you to my collaborators. Thanks to the participants. Thanks to our funders. Thank you to Stanford for giving me this opportunity for presenting. And if you don't already, please follow me on Twitter. That's all.
Thanks, Manoj.
Cool. Anybody have any questions?
Yeah, plenty of time to hang out for questions. Maybe the best way to do it is if people want to raise their hand and then just speak up themselves. I think it'd be more lively to have a real discussion.
Yeah, you can also just shout at me.
Okay, I have a question.
Please.
So what would decreases in cognitive flexibility and then overload in neural flexibility, what would that look like? As you kind of hypothesize with the bell curve?
Right. So I think patients with schizophrenia actually will have decreases in cognitive flexibility, but somebody in psychiatry, please correct me. Patients with, there was a paper from Danny Bassett, and she's like the second senior author on it. The first author's name is Braun, Braun. They looked at patients with schizophrenia or psychosis and then first degree relatives, as well as dextromethorphan. And they showed that you get in some kind of measure of neural flexibility. She used actually a different sort of approach of functional connectivity. And I want to say, I think she used a sliding window approach. But she showed that in brains of patients with schizophrenia, as well as their first degree relatives, that you get greater neural flexibility. And yet you wouldn't expect in those patients to get greater, to have greater cognitive flexibility. So I think that, yeah, at some point you could get impairments in cognitive flexibility. You also, I think one thing people have also shown, so this is one of our collaborators, Monica Rosenberg, she actually showed that that you can use dynamics to better predict, as well as static functional connectivity, to better predict attention. And what's interesting is that dynamic functional connectivity was negatively, the predictive edges were negatively correlated with attention. So too much dynamics could mean bad attention, which I think makes a lot of sense. I think there's also John Mansell's work. He's shown that noise correlations, that decreases in those happen with greater attention. So again, less variability means better attention. So I think that, yeah, you get impairments in attention, probably impairments on these types of cognitive flexibility tasks. There might be certain situations in which too much dynamics is probably a good thing, to maybe get yourself out of a kind of attractor basin, rumination or something like that, to bring you out of it. Even if bringing you out of it might be an overshoot, maybe you can come back a little bit later after, some of the effects of psychedelics have worn off. So this is all just massive speculation, by the way.
Okay, cool. Thanks.
Oh, thanks.
Please, whoever, go for it.
Cool. So it seems like part of the purported mechanism for the PTSD treatment with MDMA is memory reconsolidation. And I'm curious if any of your results here on memory encoding would indicate something, especially about repeat doses of MDMA. So is maybe the effect actually being driven by like poorer encoding of traumatic memories between sessions or something?
Right. Yeah. So in the MDMA paper, yeah, we actually speculate about reconsolidation, and later on somebody wrote a little review where they talk about that. And so what we actually say was that MDMA impaired encoding, and we saw this like hint towards impairment of retrieval. And we do report this in the supplemental material, by the way, but these models, modeling using the dual process signal detection model to get recollection familiarity, it doesn't handle false memories very well. And so what you'll see is a lot of time impairment in recollection, especially if you get really high amounts of false memories. So THC at retrieval will do this. You'll end up seeing like zero estimates of recollection, saying that basically people are amnestic, when that's not the case. Yes, people's memory encoding is bad, but they can still retrieve things when they're on THC that they encoded when they weren't on THC. And really getting zero estimates of recollection is like an amnestic, like a person without a hippocampus. So these models don't handle false memories very well. And so that impairment that we do report in that paper, I kind of almost want to take back that it, first of all, wasn't statistically significant. And we do report that false memories increased under the effects of MDMA. So the original interpretation was that, oh yeah, if you can encode and then retrieve, if you can retrieve information in a less emotional way, because we only found those effects for emotional memory, you retrieve them in a less emotional way, and then you re-encode it in a less emotional way, then this should result in a less kind of, maladaptive memory. But what might also be happening, if that is true, is that yes, maybe re-encoding that memory is now being encoded in a less emotional way. But given that we saw that there was these memory distortion effects, that there was increases in false memories when you try to retrieve under the effects of MDMA, this might be related also to greater vividness under MDMA. If you can mentally imagine something as being more vivid, you also might think that you actually experienced that. And so there might actually be a memory distortion process going on here under the effects of MDMA, that, you can distort a memory in an adaptive way, you can distort it in a maladaptive way. And so I think that could be playing a role. And I think that the problem is reconsolidation in humans has been rather difficult to really demonstrate. In animals, it's pretty clear, at least, from what my understanding. But in humans, maybe not super, super great. And so finding the right paradigm will be something to really test this with. And there's one study with propranolol that I think they sort of showed an effect, but we'll see what happens, with MDMA and or psychedelics even. And I think one idea too is that it might be difficult to destabilize memories in humans. So for anybody who doesn't know, reconsolidation is the idea you retrieve a memory and it undergoes a state that's labile, and then you can mess with it. You can delete it, you can strengthen it, you can distort it. And so in animals, it's been really easy to show, but in humans, we're like hyper-associative machines. And so even if you can like delete some aspect of a memory, you'll pattern complete and autofill what was going on there pretty easily because you have so many associations with that memory. This might be true also of a rat that hasn't spent its whole life in a cage, maybe like a subway rat. And so, I think one thing that maybe might be happening with psychedelics, again, this is totally speculative, is that you get maybe massive amounts of destabilization of retrieved memories because of all this neural flexibility. And then you now might be able to better distort memories in an adaptive way. So again, speculation, but hopefully, somebody will study this soon, or somebody will give me money to study this soon.
Yeah, that makes sense. Thanks.
Cool. Any other questions? We have a good 12 minutes, please. Yeah.
Are there any research or objective measures for this psychological flexibility that gets talked about, or is that more rooted in patients' or subjects' subjective reports or the therapist's observations of their like psychotherapeutic process? Because I can see how cognitive flexibility on a Wisconsin card sorting task is very different than viewing your life and how your habit patterns, etc., and just being more dynamic there. So I'm curious about that.
Honestly, psychological flexibility is not really a construct I know super well, but from my understanding, it tends to be kind of more subjective reports from the individual or from the therapist. And it was shown from Alan Davis that psychological flexibility from psychedelic experiences was, I think, the best predictor for improvements in anxiety and depression, although this is based on a single survey. I'm really skeptical of single surveys because a lot of times the people who fill out those are people who are really into psychedelics and they kind of know what you want to answer. This is for anybody who's going to do survey research. I don't think it's bad, but I just think like in experimental psychology, we have multiple experiments before we publish a study. I think the least number of experiments I've had in a non-drug study is 2, and in many cases like 4 or 5. And so I recommend doing that with surveys as well to really nail down a given effect, is to have multiple surveys and really chase down the effect of where that's coming from. But at least according to that one survey, I think there was psychological flexibility was a predictor of improvements in depression. What I should mention is we did not find correlations with cognitive flexibility and improvements in depression. But this is based on the grid HAM-D. But the HAM-D, it doesn't necessarily measure aspects of cognition. It's really kind of, like your eating habits, your sleeping habits, et cetera. And one thing that is true, a lot of times is that medications can improve maybe people's mood to some degree, but it doesn't improve the cognitive symptoms. So this is like a problem, for example, in schizophrenia with the first generation antipsychotics. They were improving positive symptoms and maybe some negative symptoms. I think the newer generation was improving negative symptoms better, but cognitive symptoms weren't really improving. And so people were then looking at, can we manipulate, for example, the glutamate receptor? And it's been kind of difficult. So same thing in various other disorders, depression and whatnot. It's hard to always improve cognition. So finding just another drug that can potentially do that, might be useful for certain populations. And I think one more thing I want to say about that is that even though those two things weren't correlated, it might not necessarily be that greater flexibility means greater depression, but rather maybe greater flexibility might be what you need to then get like psychological nudges from your therapist to then later on, improve your depression. So it's what you do with that flexibility might be important rather than just, having flexibility altogether, which, as I mentioned, does have some downsides to it potentially. Is that Rudy?
Yes, this is me.
Yo, what up, dude?
Yeah, what up? Great talk. It was really interesting. Nice to see what you've been working on these past few years. We're homies from grad school, by the way. All right. So I was wondering, so this is highly speculative, but I was wondering whether the kind of special effect of psychedelics you found, the enhancement of familiarity coding, whether that might contribute to the purported therapeutic effects of psychedelics on depression by reducing neophobia, reducing the aversion to behavioral change that could, improve patients' lives.
Yeah. Okay. So this is all speculative, and what I should also say is that at the bare minimum, it was a sparing of familiarity, whereas all other drugs that impact recollection also impair familiarity at higher doses. But if there is an enhancement, there might be some reasons for that. So one, as I mentioned, that semantic priming was enhanced, as was the pop-out effect. It's a very low-level visual effect. So that some of these are actually a lot of times like perceptual fluency and semantic fluency. Really what familiarity tends to be is how fluently you're processing these things through semantic and perceptual networks before they reach the hippocampus. So it's really like a novelty signal. And if things are more novel, if they're more easily processed, more rapidly processed. So it might make sense then that familiarity should be enhanced if these semantic and perceptual low-level processes are being enhanced. The other thing is that semantic and perceptual networks, those are a lot of times composed of consolidated memories, really old memories that are hard to disrupt. Whereas things that are supported by the hippocampus might be the more easy ones and they're more distortable. And so by increasing, perhaps information flow through these, cortical networks that support familiarity, there might be some room to adjust what's going on there. And so, yeah, this might be related to crystallized types of patterns of behavior or thought that might be, that you might be able to adapt under the effects of psychedelics. But again, this is, very, very speculative. This is just kind of the first pass, and hopefully we'll have, there's a couple different studies we're working on to see whether or not we can replicate this effect of psychedelics on recollection and familiarity. Any other questions?
Hi, I have a quick question, or actually two questions, if that's okay, if time allows. So the first question is, so you mentioned that the schizophrenics, for example, they had too much neuroflexibility, and therefore their cognitive flexibility is compromised. So does this imply that there's like little to no role of psychedelics in the treatment of schizophrenia, or like maybe not treatment per se, but alleviation or whatever?
Okay, well, so for one, I don't know if the neural flexibility is necessarily directly related to the cognitive flexibility. I was, again, kind of inferring that. But, as far as psychedelics and schizophrenia go, I mean, there's also patterns that happen in schizophrenia that are really hard to change, patterns of thought. And so, I imagine one day after the bipolar studies happen, after some things that are, associated with psychosis, sometimes like, various stages of Parkinson's disease, you can get kind of psychotic-like symptoms that perhaps, after those studies, start to be done, that someday somebody's going to do a study with schizophrenia and psychedelics, I imagine. And maybe they'll start with low doses. I don't know. I don't know. Maybe I'm wrong here. Maybe this won't happen in our lifetime at all. But I imagine somebody's going to try that. If you read Rebus, they definitely insinuate many times that there could be something for psychedelics and schizophrenia. Although they then do say, they have a warning where they're like, we're not saying that this should ever happen. But they do insinuate it a lot. It seems to be an underlying thing that's said, because they talk about these habits and patterns in schizophrenia that are hard to kind of break. So I don't know. I mean, I don't want to be the person to say people with schizophrenia should try psychedelic. I don't want to say that at all. And I don't want that anyone to take that away from this. And then I'm responsible for somebody having worse schizophrenia than ever. And I should also mention that I have a couple friends, both a female and a male friend, who have taken psychedelics and they've never been the same since. They're on antipsychotics for the rest of their life. They're more or less normal when they're on their meds, but a lot of times they don't want to take their meds, and then they go off the rails for sure. So yeah, I don't know. I don't want to be the first person to try that. But maybe somebody will one day when we have more data, especially when we have a better idea of how to use psychedelics for therapeutic effects. Currently, we just have people sit on a couch with eye shades on. I don't know if that's the best way to do it. I think it's one way that seems to work, but is that the way that's maximized the efficacy, minimizes the harms? I doubt it. So I don't know. Yeah, I don't want to say there's no benefit. There will be no benefit of psychedelics for schizophrenia, but I also don't want to condone that.
Do we know what types of people are susceptible to psychosis? When they're coming from no history of psychosis, like your friends?
Too much neural flexibility. No, I'm just kidding. Maybe, I don't know. Yeah, I mean, I think having a history of psychosis it seems to be something that might be associated with that. Yeah, I don't think it's super well understood. And, in all of our studies, we massively screen people. I wouldn't qualify for our studies, or I don't know if we've changed this, but second degree relatives, we for a while weren't allowing in. So, you can't, if you have bipolar or schizophrenia, you weren't allowed into our study. If a second degree relative did. So yeah, I mean, it's hard for me to say, but I think there's definitely enough instances where people, there's enough case reports where, yeah, people report getting schizophrenia or schizophrenia-like symptoms, at least for some period of time, or at least for, they have kind of after effects that still sort of persist and impairments in cognition. But I don't know. Yeah, my guess is, yeah, certainly having a history of psychosis. But I also think that if you just give enough drugs to somebody and really mess with them, I'm sure you can make anybody crazy. I think that's certainly possible for anybody, but usually you're not trying to go for that. So it doesn't happen typically to people that have, more stability. But yeah, massive speculation. I have no idea.
To that point, Manoj, I mean, there's I think there's a more clear established link between, say, amphetamine use and risk of schizophrenia or precipitating psychosis. And there may be many, many reasons for that. But again, the parallel between this disruption of a resting state network or disruption of a default mode network or some neural stability versus flexibility, which could be a non-specific drug effect, to your point, may not be specific to psychedelics.
Yeah. So as far as amphetamine psychosis and whatnot goes, so there was I forgot the first name. The last name's Kapoor. And I want to say this paper was around 2003 called Aberrant Salience. I'm sure some of the people here know what this is and probably know it better than I do. But in that paper, this is, before the psychedelic, renaissance or whatever, they do talk a little bit about how early psychosis, even before the prodromal stages, that can sort of reflect, it sort of looks more like the effects of psychedelics. That is, people will have this is when they tend to have more visuals or visual hallucinations. And then later on, those can turn into just strictly auditory hallucinations. Also, a lot of people maybe even get pleasure out of it. And then eventually, some of the delusions and whatnot, maybe they're hearing, oh, you're amazing, you're amazing, you're amazing. And then eventually those turn into go kill somebody. And, then they start to become kind of harmful later. Later stages of psychosis tend to more resemble amphetamine psychosis. So there might be a switch from, serotonin to dopamine, but I don't know where the psychosis field is at with that. I felt like that one person, she had, I forgot her name already.
Yeah, Melinda. Yeah, great. Thank you so much. Yeah, if I may just ask another question, a really quick question. Wondering whether you've done any studies regarding the recollection, metamemory, and familiarity while subjects were being dosed during retrieval on psychedelics.
The Psilocybin study was during encoding, but they did test memory like 4 hours after drug administration. So there might have been some lingering effects. I think psychedelics are probably going to look like what we saw with MDMA. MDMA increased. There was a trend towards increasing false alarms or false memories in our MDMA. That's also been shown with amphetamine, so it's hard to decouple. Why is MDMA doing this? Is it because of its psychedelic effects, the serotonin 2A effects, or is it due to the stimulant effects? But THC does this. It's also, I think this was one of Robin's first studies where they found that there's increase in mental imagery under the effects of psilocybin. My guess is you would see during retrieval, if it was strictly at retrieval, we would see increases in false memories, which, might be something that we should consider during psychedelic therapy. I know a few different groups who have done, some of their patients have reported abuse, that they're recovering memories. And if, for those of you that don't know, the whole idea of recovered memories is mostly garbage. That was a thing that was in the 90s, and then Elizabeth Loftus showed that it just statistically can't be the case that this many people have, parents who are in a satanic cult that abuse them. And so, a lot of people have reported, and I should be really careful of which, I'm not going to say which groups have told me this, but they have reported that sometimes people will say after a psychedelic experience that they had abuse, and they never reported that abuse during all the preparation and all the preparation that they had. There was no signs of it by any means. And so with, the number of times that I've seen that reported just doesn't seem statistically likely based on how often abuse seems to be there. And so I'm not saying all of those are not true, but the idea of recovering a memory is probably not a real thing. Usually, I could have shown you the emotional memory data, but it's really hard to forget emotional things typically. No matter how much you forget them, it can be really difficult to forget them. So yeah, you know, I do wonder if, I do predict that under psilocybin, we would get increases in false alarms, which, you know, we can interpret as a false memory if they're high confidence false alarms. So that's what, but we didn't do the study. So yeah, it hasn't been done yet.
Great, thank you.
I see a hand raised from, just go for it, say it.
Hey, so I have a quick question. So from my understanding, like set and setting is very important for psychedelics. Is that something that you guys take into account? Is there a way to measure that and have it, because it's got to be a variable, right? Like in your data set.
Okay, so this is, okay, so for one, I hate the term set and setting because I just think that we already had terms that existed in the psychology literature. We had context and we had, you know, state, your expectations. And now we're just kind of, you know, this is the psychedelic exceptionalism that I was talking about where, and I'm not putting this on you, this is other people's fault for putting that out there. But the other thing is that state and context, it's like, this is true of all drugs. This is again related to the psychedelic myopia. I was actually trying to think of examples earlier that I thought of them earlier in the day and then I forgot them later. This was one of the examples where it's like, really, isn't this true of all drugs? That if you give people SSRIs and you have a lot of just, you know, really good support, that's going to improve their condition. Well, Robin just showed that. If you looked at that New England Journal of Medicine paper, people found there was a pretty large improvement of citalopram 1 week after dosing. That has never been shown as far as I know. And so it usually takes like a month. And so what that says is that all that preparation had an effect. Maybe there's all that preparation plus the citalopram for a week interacted. But yeah, I mean, I think this is true of everything. I think the one thing that psychedelic research and medicine gets correct is that we really do a lot of follow-ups and a lot of preparation. This should happen across all of psychiatry. And if anything, I think it's more important with antidepressants such as citalopram because those are associated with increased suicidality in the first couple of weeks. And so we should really be following up with those people, especially more so even than the people after psychedelics. So yeah, I mean, I think that yes, these things are important, where you do the drugs, what your state was, that maybe I don't know. I mean, maybe you shouldn't be in a super anxious state beforehand, although a lot of these people are always anxious. And so I guess you should do the best to try to calm them. Certainly being in a safe space helps. But, one of the things I was trying to get at is that, is it really the case that, doing psychedelics on your couch with an eye shade on is the best way to do them? Or is there really something to doing them out in nature, for example? People have shown without drugs that walking through nature improves your working memory compared to walking through a city. So, maybe there's an interaction there. Nature has more entropy in it. There's more fractality. This might, I don't know, influence fractality and entropy in your brain, although this is, just me mapping on things that might not actually map on. But yeah, those are, they're, I don't know. Yeah, certainly they do make a difference, but we keep everything relatively constant, and it does involve a lot of preparation pre and post drugging.
Is there any type of screening system you do to kind of measure somebody's mood before giving them medication?
Yes, and I don't know what has been done with those data. I want to say there is probably something that's been published there, but the problem is, one, these are really small ends, and then if you were to look across studies, we're talking about really different populations. You have your terminally ill patients, we have the healthy people who never had a psychedelic experience before, we now have this depression study. And so how people's state is before receiving psychedelics might not be the same for one group compared to another, what it does afterwards. But yeah, I think that would certainly be, this is something we can actually do in the lab, for example. You could just give people the Trier social stress test before they, take a massive dose of psychedelics and then see how they respond afterwards, or you could give them a, more, just kind of basic, or just not do the Trier social stress test. And people are going to be stressed, and then give them psychedelics and see how that influences their trip. I think these are all really interesting things and just basic science questions that I would love to answer myself, or that I hope somebody else answers. But money is the problem.
Could I ask a philosophical question?
Please.
Well, basically the question is, what is cognition at all? I was very intrigued by the title of the talk, and I really enjoyed it. So one of my favorite topics is thinking. I mean, I guess we might want to distinguish between cognition and thinking, but just to maybe provoke or bring up a possibility, I have been working with a medieval notion of cognition. It's from Thomas Aquinas, actually. And he does a vivisection of cognition, so like a cutting open and spreading out an analysis of cognition. And the stages are, number one, there has to be a kind of breakdown and a confusion where there's all these perspectives. Then number two, if it goes right, an essential thought or intuition will kind of break through and pierce this multiplicity, and the confusion will suddenly be bathed in this light of this idea that is kind of surged forth. And then third, this intuition or idea will illuminate all of the perspectives, and the confusion will be alleviated and everything will go into its right arrangement. So, I mean, to me, it almost sounds like that whole process of cognition, that's what a trip is. Maybe the trip as a whole is a single cognition. There's a kind of confusion, and then maybe some sort of authentic idea will break through, and maybe there would only be a single cognition in a whole trip.
So I mean, really, okay, so I'm experiencing a bit of confusion, but let me answer your question of what cognition is.
Yeah.
So first of all, if you take an intro psych class, you'll learn about the history of psychology, and they'll go into Freud, where here's this random model, which, by the way, I can't believe Freudian theories are entering psychedelic science. But here's this guy who had an obsession with his mom, and then he just mapped it onto all the ideas of how the mind works. And then, it said that this is how everyone experiences things. And then, the behaviorists came along and were like, hey, this is, we're going to have observable data that we're going to make a real theory about. But behaviorism was too simple and tried to explain absolutely everything. And then the cognitive revolution happened. We're sort of treating the mind or the brain kind of like a computer with information processing. And so you have different types of information coming in that are processed in different ways. They're converted to something else. Like you take light and you're converting it into an impulse that, eventually projects to different parts of your brain. And, you eventually get this image of what's flat on your retina then becomes three-dimensional. You're perceiving it as three-dimensional. So I think it really just comes down to just information processing is really what we mean a lot of times by cognition. And so I don't think that a psychedelic trip is just one unit of cognition. And to be honest, I don't really know what a unit of cognition necessarily is. I think we arbitrarily break things down sometimes, but I think that's certainly not correct. A single unit of cognition across a whole, let's say 4-hour trip. So yeah, I mean, I think that's kind of, but I guess to your point of things being confusing and then making sense of them, I think is what you were getting at. So, one thing that, Rebus has tried to do was blending predictive processing with entropy. And so the idea with predictive processing is actually the world's not confusing because you're predicting what's happening first. And so there's always some kind of prediction, and, forget even Carl Friston's, predictive processing. You can show this with language and various other, types of behaviors. So, yeah, I don't know if I answered your question because I also, I ended up confused because I couldn't predict what your question was. So, yeah.
Yeah, I don't even know if I really had a question.
It's experiential. It's the experience of the question.
Yeah, that was pure experience. No prediction. So yeah. Any other? I see. There's the chat really just blew up here. Okay, cool. Okay, I don't see that. Okay, great. Most of these aren't questions.
If I could just shout it out.
Yeah, please.
I was wondering, so you were sort of saying that the default mode network-centric story of psychedelic effects is overplayed. And I was wondering whether you had a different sort of preferred account of the neural bases of ego dissolution.
For ego dissolution, I don't. I don't know if there's a single thing called ego dissolution, first of all. And if you looked at some of the original surveys that the Swiss group was using, they referred to it as anxious ego dissolution, whereas I don't know if the way Robin thinks of it, ego dissolution is necessarily a bad thing. It might be more associated with oneness, which I think people a lot of times think of as a good thing. And so I guess I should say there's a couple things. I was originally going to give you one answer, and then now I think I've got two. So one, as far as just basic mechanisms of psychedelic drug action, we're actually putting out a paper. It's in review for the second time because somebody wanted us to, we're talking strictly about serotonin 2A drugs. And I hope my reviewer was here because they asked me to then talk about our own paper, the Salvia paper, which we were trying to avoid talking about because I think there's too much, just kind of gratuitous self-citation, and we were avoiding that. And now I have to do that. Even though Salvia had nothing to do with what we're talking about, and it's just as relevant as ketamine, psychosis, or really any other drug. So, but in that review, we talk about 3 circuits that might be important to psychedelic drug action. And I think the one that has the most, so we try to make sense of the circuits of Rebus, which are rather ambiguous at times, because what's a low level and what's a high level is unclear. But I think what's low level tends to be sensory and evolutionarily older structures, which isn't really lower level. Your thalamus has co-evolved with the cortex, such that there's higher level regions of the thalamus. Your hippocampus also is not necessarily a low level region. It also performs lots of prediction, high level prediction. But we try to make sense of the circuits, and the circuit that seems to be, that Robin has explicitly stated in Rebus was the hippocampus, or really just the whole, the hippocampus and the whole parahippocampal gyrus, which is also not an inhomogeneous structure. It's a heterogeneous structure. And then the default mode network. That was kind of the circuit that seemed to make the most sense. And I think that there's the least evidence. There's not a lot of evidence for necessarily. The circuit that I think has the most evidence that's been shown across different labs, it seems to be shown in animal models, would be, I think, the corticostriatal thalamic cortical loops. So basically, thalamic gating, I think, might go awry, and that's now, something's happening there. And there's actually this whole idea from Murray Sherman. He's an anatomist. I don't know if he would describe himself just as an anatomist, but he talks a lot about thalamic anatomy. And there's this whole idea of efferent copies that are going on between the cortex and the thalamus that involve layer 5 pyramidal neurons, where serotonin 2A receptors seem to be mostly on. And it involves then prediction. Essentially, an efferent copy is a prediction of what your movement's going to be next. And there's this whole idea of keeping the higher levels in the thalamus informed about what's happening next. I recommend reading, it's a Nature Neuroscience paper from 2016, but it kind of actually has certain ideas of Rebus in it, but involves the thalamus and the cortex. So I think that might be the best circuit as far as psychedelic drug action, so far anyways. As far as what else could cause ego dissolution. So what I didn't tell you about how that modeling procedure works when I'm fitting recollection and familiarity is that recollection is thought to be a threshold-like process. It's all or none, kind of like an action potential. Whereas familiarity is more of a continuous graded signal detection process, if you know signal detection theory. And so one idea might be that these heuristics in the brain that are either yes, on or off, kind of thresholds are being reduced, and that more continuous sort of processing is being facilitated. And so, kind of sharp boundaries are being kind of extinguished while, these kind of continuous, more flowy sort of processes are being facilitated. And there is actually a way of showing this in perception that I would like to do one day, but I haven't been able to convince any of my bosses to be able to do it, where you can show actually that there's a continuous process in perception and a threshold-like process in perception in terms of change detection. And what's really cool is that they involve the opposite neural correlates as episodic memory. So the threshold process recollection in episodic memory involves the hippocampus. The continuous process familiarity involves the cortex. This is completely flipped with perception, where the threshold process involves the cortex and the continuous process involves the hippocampus. And this is, I'm simplifying this quite a bit. But the idea there is that rather than psychedelics modulating specific regions of the brain, they might be driving certain computational processes in different ways. And so, you could really extrapolate from this and think that, okay, if you're breaking down kind of these sharp boundaries for kind of more continuous processing, this could also be extended to other, somewhat arbitrary sharp boundaries that we put onto the world, such as the self versus other. But, this is, again, remains to be tested. Did that answer?
Sorry, I do have a question. Sorry if I cut anyone off there. But just in relation to what you found on cognitive flexibility and depression, and I guess there's sort of the presupposition, which may not be valid, that neural flexibility has some association with cognitive flexibility. But I was just wondering your thoughts on sort of the literature surrounding how psilocybin, for instance, is working on the serotonin 2A receptors. And then we've got the calcium signaling cascade, which is going to create sort of greater plasticity. And I know this is a rudimentary area of research, but I think it holds some promise. So do you think there's any association between sort of the plastic effects of psilocybin and classical psychedelics more broadly in some of the findings that you've shown?
Yeah, and so in the paper that we just resubmitted, we actually do talk briefly about that. So in humans, it's kind of difficult to measure plasticity. And first of all, a lot of plasticity, I don't always know what people mean by it, but it can mean, anything from facilitating, synapses to, the spines on synapses, and also even neurogenesis, right? Some people might refer to that as plasticity. And so one thing that we kind of describe in this paper is that, well, neural flexibility, as measured with fMRI, might be a proxy for plasticity. Although, again, I don't want to make that, I don't want to say that too strongly. So one idea is that if you're constantly having kind of de-weighting and re-weighting of different synapses happening all the time, you're going to then get differences, large changes in functional connectivity patterns. And so that should then result in more variability, which is what my measure of neural flexibility was there. So I think there might be something there. And I think the other thing that I should mention is there are people who show that neural flexibility is highly related to cognitive flexibility. Especially there's a study that showed neural flexibility specifically of the salience network, which one of the hubs is the ACC. That especially seems to be predictive of cognitive flexibility. And this is also true of, there's other studies that have shown this, I think, but what is her name that I'm forgetting? It's Lucina Udine, I think. Yeah, she's shown kind of measures of flexibility in the brain being related to cognitive flexibility. So there's that. I think I answered the first question a little bit in the last question, but I think another part you were saying is serotonin 2A receptor and calcium signaling. So I am not a, neurobiologist. I know very little about calcium signaling other than, there's some importance there with long-term potentiation, as well as this is also kind of a method of, of looking at neural activity. But at least as far as the serotonin 2A receptor goes, it might be the case that there's something else, that there's other things that are important. The 2C receptor might be really important. It seems to be the case that, ketanserin, it does block the 2C receptor as well as the 2A receptor. I think that somebody, I forgot who I was talking to recently, was saying that there's a psychedelic that doesn't really bind very well to the 2C receptor and it doesn't have very strong visuals. There's the cognitive flexibility literature in animals is a mess. And I don't know if I should say it's a mess, but it seems like it's a mess. And they'll show that, there's different things. When I say that the cognitive flexibility literature in animals, I mean revolving around the 2A receptor. So there's people who show that 2A antagonism decreases cognitive flexibility, and then that 2C antagonism might also impair cognitive flexibility, or maybe impair it, I can't remember. And then there's a study showing that if you administer one of the NBOMs, 25I, I think that this impairs cognitive flexibility, but then if you administer a 2C antagonist, it now preserves cognitive flexibility. So that there's some weird interactions between the 2A receptor and 2C receptor. And it seems like whether you block it or you activate it, you're impairing cognitive flexibility in animals. And then it seems like with the 2C receptor, there's situations in which you can get enhancements in cognitive flexibility, and other situations in which it prevents, it can also impair things. And so, I don't know if the 2A receptor, I think that, Scott Hamilton's work, what did he just show? That I think he was wanting to say that the 1B receptor might be important, although I don't know if the phenethylamines bind to the serotonin 1 receptors. I know that they bind to the 2 family, 2A, 2C, 2B, but I think the phenethylamines might not be as promiscuous, in which case that would then suggest that, then the phenethylamines might be less therapeutic, which I don't know if people would like to believe that. I think a lot of people think mescaline is quite therapeutic. A lot of people might like 2CB. So anyways, I don't know. I think you had a few questions in there and I hopefully answered some of them.
Yeah, that was great. Thank you.
Manoj, I got it. Rich, I see your hand is up. Unfortunately, I do have to run. So I got to end this.
It's okay.
I would say prematurely, but I think it's been very, very thorough. I super appreciate all the time, Manoj. So thank you for coming and sticking around. And
yeah, feel free to if you want, message me on Twitter. I'll try to get at any questions you might have. So
awesome. Thank you.
And thanks for having me and all of this and listening to me. And yeah, please, find me on Twitter if you have any questions.
Thank you so much. And thank you for hosting, Gianni.
Absolutely. Yeah. Good to see everyone. Take care.
Later, y'all.
Peace.